Hi, this is BT Ahiri with the topic synaptic transmission. Here we have focused onto the release of a neurotransmitter from the presynaptic neuron. Under this, we are going to cover chemical synaptic transmission, release of neurotransmitter from the presynaptic neuron and its relevant exercises. The learning objective is to have the basic idea on synaptic transmission, especially focused on release of neurotransmitter from the presynaptic neuron. So the first question is, what is synaptic transmission? Synaptic transmission is the conduction of nerve impulse from one neuron to another. So here we have already highlighted the synapse. The neuron which is existing before the synapse, it is the presynaptic neuron. And the neuron which is there after the synapse, it is called the postsynaptic neuron. So our focus is to see how the action potential, as it is in a wave-like structure it used to move, how it goes from one neuron to another neuron. This is the process we are going to cover over here. So why do we use chemical synaptic transmission? Because synapses are of two types. One is the chemical synapse, another one is the electrical synapse. Chemical synapse is the junction between the two cells through which nerve impulses are transmitted slowly, and the direction is one way always. So here the signal used to move in the form of chemical signal. It is generally seen in the higher vertebrates. And for electrical synapse, this is the cell-to-cell junction, obviously. And from here, rapid transmission of nerve impulses we can find. And the electrical signal, as an electrical signal, the nerve impulse used to move from one neuron to another by means of ions. In lower vertebrates as well as invertebrates, we can find this electrical synapses very much. So as the chemical synapses are very much common, so our focus is on the chemical synaptic transmission. This particular diagram is presented over here. It denotes the difference between the electrical synapse and the chemical synapse. How the differences are existing. See, in the right hand side diagram, this is the chemical synapse. Over here, the vesicles are present, and within the vesicles, these are the synaptic vesicles. Within the vesicles, there are the neurotransmitter which are existing. But neurotransmitters vesicles are not present in the electrical synapse. And if you see properly, the difference between this junction area is also varies among the electrical synapse and the chemical synapse. So there exists difference between the electrical synapse and chemical synapse structure. So synaptic transmission as a chemical synaptic transmissions include three broad phases. The release of neurotransmitter from the presynaptic neuron, which is today's focus point, and the other two are changes in the synaptic cleft and the neurotransmitter binding to specific postsynaptic receptors. These last 2 points we will cover in our next presentation. So if we focus on the release of neurotransmitter from presynaptic neuron, we need to know the structure of synapse, then the synthesis, packaging and transport of neurotransmitter molecules, as well as the release of the neurotransmitter molecules. The first is the structure of synapse. See here, according to the structure, synapses are 4 types. And first one is the axodendritic, means presynaptic neurons axons and postsynaptic neurons dendrons are over here. Second one is the axosomatic neuron. And what happens over here? Presynaptic neurons axons and postsynaptic neurons soma, these are over here. Now, the third one is the axonic synapse. What happens? Within the axonic synapse, presynaptic neurons axon and postsynaptic neurons axons both used to form the synapse. And the fourth one is the dendrodendritic synapse. Over here, both of the presynaptic and postsynaptic, both of the neurons, dendrons are over here. This is the synapse. Types of synapse not only classified with the basis of structural level, but in some other level also. Here, depending on the site of the neurotransmitter release and site of neurotransmitter reception, we can divide neurotransmitter into two types. One is the directed synapses, another one is the non-directed synapses. The diagrams are always very important in the physiology part because everything can be clearly explained with the diagram. So if you write very little but give proper appropriate diagram with the leveling thing, then it is very helpful for anywhere and everywhere. So directed synapses, the site of neurotransmitter release and site of neurotransmitter receptions are in close proximity. This is the difference between directed and non-directed synapse, because in non-directed synapse, it's in distance. The site of neurotransmitter release and the site of neurotransmitter receptions are in distance. And non-directed synapse is also known as string of base synapse. This structural description of chemical synapse is very important to understand the overall concept over here. This yellow portion is the presynaptic neuron and this pink portion is the postsynaptic neuron part. Within the presynaptic neuron, there is the mitochondria. It is very important because it is the energy providing part of the neuron. So mitochondria in the terminal of presynaptic neuron provides energy to the overall process. There is microfilaments. It's the smallest fibers that help in cell movement. And there are also microtubules. Microtubules are long hollow cylinders made up of tubulin protein units and large fibers. There is the existence of vesicles. Within the vesicles, there are neurotransmitters. Here in this diagram, the vesicles are clear. Active zone is very important for this overall process because the active zone, these are the specific thickening area in the presynaptic neuron from where the neurotransmitter releases. And again, postsynaptic density is very important part. It is in the postsynapse. It is in the postsynaptic neuron. It is the concentrated area. In the second phase, we are going in the synthesis, packaging and transport of neurotransmitter molecules. Here the vesicles are of three types. One is the small clear core. Second one is the small dense core and third one is the large dense core. So from small clear core, acetylcholine, glycine, GABA means gamma amino butyric acid, glutamate, these are all secrets. From small dense core, catecholamine secrets. And from large dense core, you can say the neuropeptides used to be secrets. So types of neurotransmitter molecules are basically two types. One is the small molecule neurotransmitter and another one is the large molecule neurotransmitter. In the small molecule neurotransmitters, they synthesized in cytoplasm of the terminal buttons and packaged in synaptic vesicles by the buttons Golgi complex. Vesicles with neurotransmitter are stored in cluster next to presynaptic neuron. The sizes are smaller. For large molecule neurotransmitters, the size of the vesicles are large. And these are basically the peptides, amino acid chains composed of 10 or fewer amino acids. These neurotransmitters are assembled in the cytoplasm on ribosome, packaged in vesicles by Golgi complex and transported by tubules to the terminal buttons at 40 centimeter per day. Earlier researchers found that the small molecules neurotransmitters are only in the presynaptic neuron, but for that neuron there is no existence of large molecule neurotransmitters. So in the presynaptic neuron, either small molecule neurotransmitters existing or large molecule neurotransmitters are over there. But later on researchers has found that both type of molecule neurotransmitters can be existing within a particular terminal end. Third phase, the release of neurotransmitter molecules. The process of neurotransmitter release is known as exocytosis. This process is of two types according to the size of the neurotransmitter. For small molecule neurotransmitters, it is typically released in a pulse each time an action potential triggers a momentary influx of calcium ions through the presynaptic membrane takes place. So we are going to cover influx of calcium ions in our next slide. Large molecule neurotransmitters are basically peptides that we already knew, are typically released gradually in response to general increases in the level of intracellular calcium ions, such as might occur during a general increase in the rate of neuron firing. So here is the role of calcium, which has been clearly given. So over here, what is mainly important, when the action potential reaches in the terminal button, some events are very important. Within it, the first thing is there exists voltage gated calcium channels in the presynaptic neurons. And whenever the action potential reaches, these presynaptic calcium channels used to be open. So calcium enters into the intracellular fluid from the extracellular fluid. So calcium comes from the outside to inside, and that increases the calcium level in the intracellular fluids. After that, calcium binds up with the vesicles. Vesicles comes pre-synaptic membrane and it helps neurotransmitter to release after that. Here is the role of proteins. There are basically two types of proteins we can see. Vesicles protein is called the v-snare protein. It is also named as synaptobrevin protein. See, over here the vesicles are mentioned and along with it the v-snare protein is there. In the presynaptic protein, it is called the t-snare protein. This is the presynaptic membrane, and over here the t-snare protein is existing. It is also named as syntaxin or SNAP25. It helps in docking. Docking is a process that attaches vesicles to presynaptic membrane, and it is also helping in the priming process. What is priming? Priming makes vesicles ready to discharge their neurotransmitters. So action potentials in this particular diagram flows from here to here. Now interaction between the v-snare and t-snare lead vesicles to attach in presynaptic membrane and helps in releasing neurotransmitters. That is very important part. While vesicles attach with presynaptic membrane create a hole through which neurotransmitter releases. After releasing, resealing takes place. Vesicles go back to its initial place through endocytosis. So this is the entire process. Vesicles come down and go back. This process is known as case-run discharge or case-run process. Here in this diagram, the docking, priming, releasing of the neurotransmitters from the vesicles is shown through fusion. And after that, this area used to be covered up. Resealing takes place and the vesicles goes its initial position. This is the step of synaptic transmissions. The first 6 points are already covered in this presentation. The last two part we will cover in our next video. Here is the brief overview about the states of changes in the presynaptic neuron. When the action potential reaches to the presynaptic terminal, what are the points we need to cover in that particular phase? First one is the structure of synapse, and within it we have already gone with the structure and description of chemical synapses. Secondly, the synthesis and packaging and transport of neurotransmitter molecules. Within this point, we have already gone with the types of vesicles, types of neurotransmitter molecules. And finally, the third point is the release of neurotransmitter molecules. The entire process keys and run discharge. Within it, we have already covered exocytosis and the role of calcium and protein over here. From this discussion, you can go with these questions. Define synaptic transmission. Differentiate between chemical and electrical synapse. Types of synapse. What is exocytosis, docking, priming. And you can also able to write a note on structural description of chemical synapses. You can also able to discuss the role of calcium and protein in synaptic transmission. If your concepts get clear with this particular presentation, you can like, share, you can give comment and also subscribe this particular channel. Finally, it's thank you for your active listening.